Well, welcome everyone, and thanks for joining us for this first Arthur Rylah Institute seminar for 2025. It's really great to have people back who are returning from last year, but also welcome to any people who might be joining us for the first time today. I'm James Todd. I'm the Chief Biodiversity Officer and ARI Director, and I'll be chairing today's seminar with technical support from Andy and Tess. I'd like to begin by acknowledging the traditional owners on the land I'm joining you from, the land of the Wurundjeri people, and to pay my respects to their elders past and present. I'd also like to extend that acknowledgement and respect to the traditional owners on the lands that you're all dialing in from today, and to any traditional owners and Aboriginal people who might be joining us online today. Today I'm really excited about our special guest presenters. We've been very kindly joined by a couple of presenters all the way from Western Australia. We've got Associate Professor Barbara Wilson and Honorary Professor Mark Garkaklis. Both Barbara and Mark have a wealth of experience working as ecologists in both government and academia. Their expertise and research have considerable overlap with our work, and today they will be addressing a topic of great importance across the environment sector, and that is Phytophthora, otherwise known as cinnamon fungus, and its impacts and management. So starting with Barbara, Associate Professor Barbara Wilson has conducted long-term research of native mammals and the impacts of threats, including the pathogen Phytophthora cinnamomi, revealing significant declines in communities and threatened species as a result of its impact. She has extensive experience in government and academia in Australia and internationally, including in state and Commonwealth conservation legislation, policy and strategy. And Barbara is well known to many of us, having been previously an academic in Victoria and part of the Scientific Advisory Committee at some stage. So today, Barbara will share the latest updates on the Wild Otways Initiative to protect our threatened plants and animals from the impacts of Phytophthora, and I'm really looking forward to this, and from the presentation from Mark as well. Just a note, we'll have time for some questions at the end of each presentation, so please write these in the chat and we'll try to get to as many as possible after the presentations from Barb and Mark. So thanks everyone for joining us. Thanks for Barbara and Mark being here. Over to you, Barbara. Thanks.
Thanks very much, James, and thanks very much to you and to Arthur Rylah for hosting us. So in this presentation, I'm going to be presenting on behalf of the team that was involved in the Wild Otways Initiative, which was a 3-year federally funded project. And I'm going to be talking to you about the project on Phytophthora, which was protecting and conserving threatened species from Phytophthora dieback across the Otways. So the major project conveners, managers, and the people that led us were Mark Garkaklis and Kat Lovett and Jess Miller. So thank you very much to them. I'd like to acknowledge that traditional owners in particular on whose lands this work was undertaken. So the Gabanun people and the Wadawurrung people, paying their respects to their ancestors, elders past, present and emerging, and also their contribution to the project. So the Wild Otways project, the Wild Otways Initiative, was a $6 million project that funded a range of projects on pigs and deers, fox and cats, rewilding, small mammals, and some community environment. So our Phytophthora management project fitted within that remit. Well, what is Phytophthora cinnamomi? It's a oomycete, a water mold. So it's very closely related to fungi, but it has some differences in how it reproduces. So the main invasive aspects of the pathogen are these zoomycetes that are dependent on water, and they are carried through the water. So wet areas are very important. They infect the roots of the species that are susceptible and invade them. The mycelia can work through the plant and stop it from being able to uptake both water and nutrients. And it also has another invasive pathway where if conditions are bad, that forms chlamydospores that just sit there in the soil, and when the water comes along, it will re-establish itself. So the symptoms, many of you will have seen this, are rotting of the plant, root and collar lesions, the leaves are chlorotic and yellow before they die, and there is a wide host range in Australia of native plants. The dispersal, the autonomous that I've spoken about, these zoospores and the mycelial growth, and then the passive, which is really important. That's where the water comes in, or we are moving plant material or infested soil, and recreation, road construction, all of these types of things. So it is one of the world's 100 worst invasive species recognised by the IUCN. And the epidemic of dieback is a major concern in Europe, USA, Mexico, New Zealand, South Africa. And the consequences, further than what I just spoke about, the plants are unable to develop shoots or flowers. You can get extinction of populations of flora species, invertebrates. A major and dramatic modification of plant community structure and composition is a real threat. And there is significant reduction in primary productivity and functionality. We don't understand all of that. Habitat loss and degradation for dependent flora and fauna. At the moment, it's to date irreversible. There's likely to be significant loss of genetic diversity and major declines in particular fauna species. So dealing with the problem, we have limited management options available. We can prevent the introduction or limit the spread into uninfected areas. This involves things like mapping where the infestation is, restricting sites, establishing quarantine, and improving our hygiene procedures to minimise spread, spraying down vehicles and watching and cleaning our boots. We can reduce the impact at infected sites, but the only thing we have available at this stage is the phosphite, which increases plant resistance. And that has been used long-term and wide-scale in Western Australia, and Mark will talk about that. So dieback threatening processes are listed twice under the Victorian Flora and Fauna Guarantee Act. Firstly, in terms of infected gravel and construction of roads, and secondly, from infected sites into parks or reserves under the control of the state or government authority. It's also listed under the Commonwealth Environment Protection and Biodiversity Act. And under that act, the National Threat Abatement Plan, which the most recent version is 2024, and this is currently under review. The objectives of that plan are to identify and prioritise production of biodiversity that is impacted, reduce and mitigate the impacts, inform engage the community and inform their actions, and to promote any research that may help us improve our management and protect assets. So this project was conducted in the Otway Ranges, which has a range of vegetation communities, being 100 kilometres from Melbourne on the southwest coast, ranging from heathlands, which I've circled here, rainforests and wet communities, and some major national parks. The background of the pathogen in the Otways was basically in the eastern Otways. It was observed in the 1970s and research began. Between 1989 and 2015, we had one site where the uninfested area was reduced by 83%. So we had a long-term management monitoring. And there were significant declines in the total number of plant species, cover abundance of things like grass trees. You can see this pathogen movement here, very obvious. and significant impacts on mammals such as eastern pygmy possum and white-footed dunner. So the Wild Otways program wanted to work out what was going across the Otways, and that was conducted in 2021-23. The major objectives were to identify these protection areas, prioritise protection areas, particularly with relationship to a range of listed EPBBC acted species. Another was to minimise the pathogen spread, to develop some standard operating procedures with those land managers, and to undertake aerial and hand spray of phosphite. We also had an objective to conduct hygiene training for the land managers and community groups, and to determine the current extent of infestation, although I won't address this and I'll leave that to Mark later, and to do some research on phosphite toxicity and habitat restoration. So to do the identify our priority areas, we involved all the key land managers. We identified the distribution of our threatened species and the threatened habitat from the modelling. We had a relative threat of Phytophthora, which was scaled according to the intersection of suitable habitat and proximity to roads. So these red areas much more likely to be impacted. And we used a structured decision-making process to determine the areas. So these are the areas that we came up with, 7 areas, 1, 2, 3, 4, 5, 6, 7. This is in the Eastern Otways, and this is down in the Carlisle Heath. So those areas were identified as having high level biodiversity and for us to address. So within those priority protection areas, we identified some really high-risk areas for application of phosphite. And we did on-ground spraying in the Carlisle Heath and in the Eastern Otways. We also did several aerial spraying areas, one in the Eastern Otways here at Denham's Track, and the other one on Egan's Track in the Carlisle, south of west of the Carlisle Heath. Our phosphite training, we did theory and on ground to all the land managers and some community groups. And to assist us, we introduced people from Western Australia. For example, these qualified people from Glevin, a group in Western Australia that actually have Phytophthora management as their main objective. Our phosphite application, this is an example at one of the sites. So this orange areas here, these were hand sprayed. This is a track, and you can see all these spoon drains where the pathogen occurs. And the aerial application was done just in this area here along the track. So we don't spray the whole area, we spray a barrier. Our hygiene training, we had in-field training sessions for the land managers, traditional owners, community groups, and the CCMA produced some wonderful video material. All of this is available in the links. Our research was mainly focused on rehabilitation through Deakin University, and you can see this is a site that's been heavily impacted, so a highly modified vegetation. Campbell, our PhD students, selected seeds grown by local nurseries. Plantings have occurred, experimental plots. Some have been treated with phosphite and others not. And this is being monitored for up to 4 years. So I think this has given you an introduction to the project and how you might roll out things in other areas. I think the key to the success was the wonderful collaboration across tenure and interagency. We actually had members from each agency within the Phytophthora program. We had experts coming from Western Australia, people like Sarah Barrett, who's worked for 40 years on the pathogen in Western Australia. Community and our traditional owners actually combined to produce some wonderful outcomes, in particular to address their iconic plant species such as grass trees. So both they hand sprayed these iconic grass trees, and they're on the way to protecting their country through their country management plan. And just finally, I think the investment of $400,000 over 3 years and the outcomes was well worth it. So thank you very much.
That's great, Barb. Thank you for a fantastic presentation. I think we did well to get through the connectivity issues there, because I think your video is not is freezing a bit, but we saw the presentation, so no drama about that. Maybe on that point then, and we've got some questions in the chat, but you mentioned traditional owners. How do you see the role of traditional owners, land management in terms of Phytophthora dieback going forward?
Thanks, James. I think they're extremely important. They've been contributed already to this project and have got their own program now of spraying, in particular, the grass trees, which are iconic for many of the traditional owner groups in Eastern Australia. And they were extremely enthusiastic. They undertook their training and have worked out their own program. And in Western Australia, we've had a great interest from particular groups that have contributed. And also going forward, the National Threat Abatement Plan, in the review, we in particular are looking for traditional owner groups to contribute to that, because they were not mentioned in the original plan. So that needs to really be improved, and they are a wonderful source.
Fantastic. Okay, well, we've got plenty of time for questions. So I might hand over to Andy just to talk through the ones we've got in the chat. Over to you, Andy.
Thanks, James. Yeah, keep the questions coming. Look, we've got plenty of time, which is a wonderful situation to be in. The first question for you, Barb, is from Daniel. He was asking if there are multiple types of Phytophthora species that cause dieback, and whether or not in the Otway situation it's just the cinnamomi, however you say that word. And he was just saying he's noting in his area that there are indicators of dieback, but he's not got any cinnamon fungus in his area.
Yes, there are different species of Phytophthora across Australia, and they've been well identified by our colleagues at Murdoch University. And they have distribution maps of the different species. In Victoria, it's predominantly Phytophthora cinnamomi. I guess the important thing is that no matter what species it is, the management is the same, okay? So that you would still prevent and use the same hygiene to prevent movement of the pathogen. And the other thing was, if Daniel has tested the soil and hasn't had a positive, sometimes that is a very difficult thing to prove, and it depends on your soil sampling.
Sampling is problematic. It's really difficult, and being able to target plants that are showing early symptoms, bulking up your samples, and then splitting in a way that's not biased is going to give you, so you've got lots of fine roots in the sample, that's going to give you the best chance of detecting it. It's a lot of false negatives,
That's a really good sort of lead into this next question that just popped up. So you're saying it's a lot of false negatives, but how easy is it to identify the fungi that might be causing the dieback? What techniques do you use to identify what's the cause?
It is difficult. You need laboratory, accredited laboratories, so that's very unfortunate. In Western Australia, their state laboratory, they have a state laboratory that does all of the identifications. We don't have that in Victoria, so we rely on universities and sending samples away. So no, you can't just take it home and have a look at it. So it does require, there are simple techniques that have been developed and used over the years where you grow up the pathogen. Now there's also DNA, eDNA techniques, so they're not something that you can do easily. Yeah.
You also touched on the last question about the management and getting onto it earlier. And it's the same thing for any Phytophthora species. Prior to spraying phosphite, what do you do? What sort of mapping or field observations or baiting programs do you do prior to applying that application of phosphite?
Yes, well, in Western Australia, over a 30-year period, they've been mapping, for example, in the Stirling Ranges, where they have high levels of threatened endemic plant species, and they have those all mapped. In Victoria, when we were doing the Wild Otways, we did some testing of the pathogen, and we did some limited mapping because we didn't have we were doing the mapping in parallel for this project, so we hadn't finished it, but we do have the on-site mapping of the extent of the pathogen moving down a hill, for example.
In Western Australia, it's a great question. In Western Australia, they've got very good systems in place. It's really managed by the Sustainable Forestry Group, who got a very structured process. And if there's operations that are taking place within natural bushland in Western Australia, within 9 months, you've got to get a dieback interpreter to go out and actually identify any hotspots or areas of the pathogen so that those operations can take place with full knowledge of where the disease is and where it isn't. In the Wild Otways, we used the same approach. We went and looked at symptomatic monitoring of the presence of the pathogen. Our PhD student had pretty well confirmed most of those areas that we actually think we've got the pathogen, it's there. So that we were actually identifying where active disease was within a few months of actually going in and doing some phosphite treatment.
Following up around the question you had about the testing through an accredited laboratory, someone's mentioned that they've got kits online for like $70 to test. Would you suggest they're not that effective and you're better off going through the accredited versions for confirmation?
I take a precautionary approach. If you have grass trees and they're dying, until you know better, you've got Phytophthora cinnamomi. Imagine you don't get it right and you have a false negative. So I don't know how many negatives you'll get from the testing kits. It's all about the sampling. It's actually the sampling, not necessarily the kit. And that's the tricky things. If you see grass trees dying on your property, until you know better, I have a grass tree on our place on the Surf Coast. I spray it every year with phosphite. I spray that. You can buy it from Bunnings. We spray it every year. We love that grass tree.
So this is a question, Bob, around the practice of spraying, and a few questions have now come around, like are there negative impacts of phosphate control, but also how do you even apply it to grass trees and what's the impact on the grass tree? Is applying phosphite safe? And how do you do that in practice?
Look, I think we should leave that to Mark because that's what he's going to talk about.
Perfect.
He's going to talk about all of the what we're going through now. That's what he's going to address. So
yeah. I would say it's a good time to perhaps synthesize that to bring us to the point where we've now measured over many decades what the impact of the pathogen is. Absolutely devastating. In many ecosystems, if you let it run, it's all over Red Rover. The idea of conserving many of our threatened species was likely to be a waste of money. So when we're trying to treat within the bush, we're not treating all of the bush, we're trying to protect one narrow bit. So far, over decades of monitoring phosphite treated areas in Western Australia, I've looked at the data. It's almost minimal, any non-target effects. I actually can't see any, but we will talk about that a bit more.
Thank you, Mark. Barb, you showed in one of your photos that you had a spray line, like an aerial spray line, to create a barrier. Do we have much evidence or information about how effective those barriers are in preventing the spread of dieback?
Yes, in particular, we have trialled it in Victoria in the Eastern Otways, and although it wasn't continued, we have been back to those sites and seen that they're looking very good, but that wasn't so scientific. In Western Australia, Sarah Barrett's work, again in the Stirling Ranges, has shown how effective it is in preventing the spread and in protecting those threatened, they're critically endangered plant species in the Stirlings. It's protected them. Not only that, she's got evidence that areas that have been sprayed, those plants are actually regerminating in the sprayed areas.
In the areas that have been impacted by the pathogen, she's getting reinvasion of Phytophthora sensitive species back into those areas that have been impacted by the pathogen. And so that's really powerful evidence.
Great. It's really good to hear. Sounds like there's a positive trajectory with the right treatment.
It's not only, just to emphasise, particularly in Victoria, it's not just the threatened plant species, it's the community structure. Because in Victoria, in many areas, you lose the grass trees, so that's vital habitat for the fauna, including mammals and invertebrates. And you get, in some places, you get bare areas. In other places, you just get sedges, and that's completely simplified. So in some areas we work in, people think that this is a lovely community, and we have to say, well, yes, but it was never like this before. It was much more diverse and had much more structure.
Mm-hmm. Well, that's good to know. Thank you. Barb, you mentioned that those priority areas for the Wild Otway Initiative. This question comes to the scaling of monitoring required to look through large areas. Are you using remote sensing data, or how did you tackle that problem of a very large area and where dieback's occurring?
Yes, Mark again can address that with how we went about it for the Wild Otways. So we did use some, you can use remote sensing, but they used high resolution photography in the end. So it's an evolving area. So I think maybe you're going to talk about that.
Yeah, briefly.
Yeah.
All right. Well, I'm just scanning for any other questions that we haven't flagged as we'll address later. I think we've got through most of them. So maybe what we'll do is I'll throw back to James to introduce Mark, and we can take questions for both of you again at the end.
Thanks, Andy, and thanks, Barb, again for a great presentation, and obviously generated lots of questions in the chat, some of which relate to phosphite and Mark's presentation. So I'd now like to introduce Professor Mark Garkaklis, who's an ecologist, and his recent conservation projects span fire and threatened species management, monitoring and evaluation, FSC auditing, and threatened process management around Australia. Mark's work in Phytophthora dieback management is directed towards habitat protection rather than individuals per se. Mark is an honorary professor of practice at Murdoch Uni in WA, and Mark will be discussing the national registration of phosphite to control Phytophthora dieback and the National Threat Abatement Plan. Over to you, Mark. Thank you.
And thank you very much, James. My name's Mark Garkaklis, and I've been fortunate to be able to work with colleagues in DECU over the last 18 months on developing a roadmap for on-label use of phosphite to control Phytophthora dieback in bushland in Australia. Briefly, I'll talk about the roadmap, what we did. Within that, there'll be some talk about Phytophthora cinnamomi and Phytophthora dieback, because it is about the pathogen that provides us clues about how we actually manage the disease that it causes. A little bit about phosphite and how it works. There is allowable use of phosphite in Australia already, and it has been used for a long time, not in natural bushland where we need special permits, but within agriculture. Why is it not currently allowable for on-label use in the bush? I will talk briefly about some of the main issues that were seen when we did our reviews to develop a roadmap for national registration. The roadmap was stage 1 of the project, and we've actually been fortunate enough to be involved to help out with stage 2, which is our actual development of a registration package to allow us to use phosphite in the bush. And I'm going to introduce you to a couple of sites that we've already been able to confirm that we'll be doing some studies at. One of those is Egan's Track in the Otway Ranges, and we're standing on the shoulders of the Wild Otways Initiative and the work we did with Deakin and Parks Vic down there and the CCMA. The other site is Boulder Hill, and it would be fair to say we're standing on the shoulders of Sarah Barrett on that particular one. And her colleagues. I know she'd be very embarrassed if she's watching. The DQ National Registration of Phosphite to Control Phytophthora Dieback in Natural Vegetation Roadmap project. The introduced plant pathogen Phytophthora cinnamomi has the second greatest impact to Commonwealth listed matters of national environmental significance, plants, animals and ecosystems. Its impact on EPBC Act listed species is much greater than introduced foxes and feral cats, and the ongoing threat from Phytophthora dieback disease caused by the pathogen is extreme. In the 2001 State of the Environment report, or was released in 2001, there's a telling graph which looks at the number of listed EPBC Act listed species impacted by introduced threatening processes. European rabbit sits at number 1, Phytophthora cinnamomi sits at number 2. The pathogen is now widespread. Despite our best efforts at hygiene, the pathogen has got away from us in many areas of Australia. We had a question about the use of remote sensing. Remote sensing is our friend when it comes to understanding the distribution of the pathogen. And I would argue that any group that's got the capacity just to look at time series NDVI derived from our historical Landsat archive, coupled with some of the European Space Agency data, coupled with any aerial imagery that you've got, will give you a good idea of where you're sitting in terms of the distribution of Phytophthora dieback disease. That little figure on the left-hand side of this screen is Carlisle Heath, a big section of Carlisle Heath, the heathlands and wooded heathlands that had been identified as a priority protection area for EPBC Act listed species. And there we're talking about long-nosed potteroo, we're talking about southern brown bandicoot, we're talking about swamp antechinus, we have habitat that's suitable for smoky mouse, and not far away we have a plant species called Taulostelia. All of them, either their habitats are significantly impacted or with the plant, they're directly killed by the pathogen. The colours on the left-hand side are little polygons that were derived from interpretation of 7.5 centimetre resolution, high resolution aerial imagery that was captured by forest fire management Vic teams down in the Barwon district. And they were able to go along and identify where the disease was occurring in the Carlisle Heath and where they saw very little evidence of the pathogen. And the red areas represent diseased or post-diseased states of Carlisle Heath. The lighter greens mean that there may very well be some evidence of dieback, but it's very low, and it was really telling to see that there was only one patch in the very south and to the west of that whole mapping exercise where they were confident, and I'm confident, that there's no pathogen whatsoever. The pathogen in Carlisle Heath is away. Given that it's already widespread, the only tool we've got at the moment, doesn't matter how much we clean our boots, the only tool we've got to limit the spread of dieback disease is the use of the agricultural chemical phosphite. How does it work? Well, the way we manage Phytophthora is according to the disease triangle. Phytophthora dieback disease will be expressed if you have host plants, and there's hundreds of plants in Victoria that are susceptible to the pathogen. If you have the right sort of environment, warm, moist soils are really helpful. Getting any autumn rain, drought is probably going to lead to less spread of the pathogen. However, if the plants are under stress because they're fighting the disease off, then drought will often kill them off, and that it'll be the last one. I'm getting a hurry up here, so that's good. And what you need as well is the pathogen. If you have host plants, pathogen, and the right environment, you'll get expression of the disease. You'll get Phytophthora dieback. Most of our focus for management has been on hygiene. It's to try and remove that pathogen element out of the disease triangle, and thereby we won't get the disease. In areas where that hasn't been effective, the pathogen is there, we've got host plants, and we've got the right environment, we're going to get the disease unless we can do something. The way phosphite works is to actually increase the plant's resistance to pathogen infection. What we're trying to do, what our management objective to do is at this stage, is to remove the host plants. By spraying plants with phosphite, it acts in two ways. It acts by inhibiting the growth of the pathogen, it slows it down, but it also acts by stimulating plant defenses. We're sort of removing the susceptible plants out of the equation. And if we remove the host from that disease triangle, we will not get expression of the disease. The way that the phosphite works is that when it's sprayed, it enters the plant via leaves. Phosphites absorb rapidly, and it's translocated through the vascular tissue. In the end, there's highest amount of buildup of phosphite in plant shoots, root tips, fruits, and seeds. It can be applied in a number of ways. According to Victorian Chemical Regulation Authorities, you actually don't need the type of respirator that Campbell has got on the left-hand side, but being a very diligent student, Campbell is hand spraying some grass trees to look at phytotoxicity responses, of which he hasn't been able to measure any, by the way, to some grass trees on a mountain bike trail in Umarella in the Otways. The slide, I love the slide on the right hand side. That was the Wild Otways Initiative program. There's a rig that was bought by that Australian government investment, and that rig is solely for the use of phosphite and applying that to protect high value habitat, and it's also there available for traditional owners to be able to protect areas of cultural importance. And this was a fantastic day where we had phosphite spray training with Wadawurrung traditional owners in the Eastern Otways. Trunk injection is really important for iconic plants that are susceptible to the disease. If you have parks and gardens where there is an objective to maintain the amenity with trees that are susceptible to the disease, then trunk injection is really useful. And in Western Australia, it's used a lot by friends groups in protecting jarrah trees and other susceptible trees in natural bushland in local governments throughout Western Australia, where friends groups have actually taken over the management of Phytophthora dieback. And also bunya pine in Queensland is managed using phosphite applications with trunk injection. For habitat protection, to me, this is the holy grail, which is aerial application. And the plane is one of your high valued water bombers that have been out working so hard in the Grampians and Little Desert over the last couple of months. In this case, we've co-opted that plane to spray phosphite on Denham's track in the Eastern Otways, and just a fantastic way and cost-effective way to be able to to dampen down pathogen expression in areas close to tracks. Phosphite has been demonstrated effective in control of Phytophthora dieback in Western Australia. I'll talk about our South Coast colleagues in Western Australia and the programs they've been involved with for decades, many decades. South Australia, Kangaroo Island is an area of high focus there. And in Victoria, and Mick Abberton's work back in the late 90s and early 2000s, coupled with work that Dave Cale and Barb did in the Eastern Otways, has given us enough evidence to know that it's a really effective tool. Phosphite use is widespread in agriculture, horticulture, and the nursery industry. Ask yourself if you've been eating avocado. If you eat avocado, the only reason you can do that is that avocado trees in Australia are sprayed with phosphite because avocado trees are highly susceptible to Phytophthora cinnamomi. If we didn't have phosphite, we would not have an avocado industry. They also sprayed on citrus and roses, and in some instances pasture itself. Its use in natural vegetation is limited. And the reason is simple. Although the chance of an unmanageable risk to the environment are very low, we've been monitoring this for a fair bit of time, decades, there's only been a moderate amount of study in native vegetation, and most of that's from WA. This means we don't have what's called an on-label APVMA, Australian Pesticides and Veterinary Medicines Authority approval for on-label use in native vegetation. We do have permission to use it, but we have to actually get that permission on a case-by-case basis or a jurisdiction-by-jurisdiction basis. That is onerous. It's onerous for states to have to continue to get off-label permitting, and there's additional administrative challenges and costs involved in that. The federal government recognised that as being an issue, and they thought, well, let's invest in this to try and remove that burden from state jurisdictions. And that's how we've got the phosphite registration for management of Phytophthora cinnamomi projects happening. The first step was to develop a roadmap for whether or not, for how we'd go about doing this, and indeed to identify if there were any showstoppers. Is there anything within all of our published literature and all of our monitoring reports that said, we're not going to get national registration? And the second step is to actually put together the data and the studies that might need to be compiled to support existing information, to put together a registration package with the APVMA. Stage one was the roadmap, and that was a review of the extensive literature that's there. We had significant stakeholder feedback from all around Australia, including the Northern Territory, which is important. And when we put together the review and looked at the available information, there was no significant barriers to national registration for phosphite. That included consultation with the APVMA on the information that we already had. There's no detrimental effects of phosphite that have been identified, while the degrading impacts of Phytophthora dieback disease have been demonstrated on flora, fauna, habitat, and for the distribution of vertebrate and invertebrate fauna. It is a degrading disease. If we let it run and it's already out there, the likely outcome is bad. It was agreed on stakeholder feedback, and I think that it is a pretty fair position that there are some additional studies that we need to understand as part of the registration package. And those really relate to the soil biome, and we're talking about using a salt acid, which does act as a fungicide and is registered as a fungicide. Is there any detrimental effects on the soil biome, the organisms in the soil? And I think that the main focus from the APVMA's point of view is they need to understand what that might be in relation to mycorrhiza. And the other area of I guess, caution is with regard to aquatic systems. And the important thing to understand when we're asking these questions about additional studies, and how we do them, is that can we identify any information that suggests if we use phosphite to control dieback disease, are those risks to the soil's living organisms and aquatic systems manageable? If we've got risks, there's always risks with everything. Are they manageable? Because if we can manage them, then that's a real go-ahead for us, because we know what happens if the disease runs. We can't manage biodiversity conservation then. Stage 2 has been invested in by the federal government, and DTS registration consultants who are specialists in registering agricultural and veterinary chemicals with the APVMA have been awarded the contract. And our project managers, Barb and myself, are actually been hauled in on that particular project with a minor part. But our project managers cracking the whips are Gavin Hall and David Dwyer up there in Queensland. The two major components of the studies are to examine differences in if any, in soil biodiversity and phosphite treated versus control areas using an eDNA approach on the soil biome, that is. And our team leader there is Jeff Powell out of the University of Western Sydney, and test any differences in terms of phosphite use on the aquatic community in control versus treatment areas. And our team leader there is Associate Professor Vanessa Gorecki up there in University of Southern Queensland. We've got a couple of sites. The project's actually officially been going for 5 weeks now, but we've moved a lot very quickly. We have gone to the areas where a lot of work has already been done straight away and lined up sites that are going to provide us a wealth of information, because number one, they are well managed with really strong systems in place. They are pre-phosphite treatment areas within those areas. We've got great support from district managers and staff on the ground. Those areas that we have locked in are the Otway Ranges, Egan's Track, which was one of the priority protection areas as part of the Wild Otways Initiative project in the Otways. And in Western Australia, a site called Boulder Hill, which is actually managed by the City of Albany on the south coast of Western Australia. Really, all of the management there is guided by the Department of Biodiversity, Conservation and Attractions, Parks and Wildlife Service out of South Coast region, which is based in Albany. I've got great support from regional manager and our project liaison with Sarah Barrett and Greg Freebury and Dion Utpa. And I would pay special mention to those three individuals about the leadership they apply with Phytophthora dieback management for all the rest of us in Australia. So they're important names to see. We've got good negotiations happening with New South Wales at the moment. Really, we're thinking that Barrington Tops is providing an opportunity, but there are several other sites that are a possibility in New South Wales, and Queensland at this stage is looking at Bunya Mountains. I've just got a couple more slides to go through, Andrew. I see I'm at 10:53. Egan's Track in the Otway Ranges, this is actually one of our sites, and this has been post a fuel reduction burn, about 9 months post burn. We've got Eucalyptus willisii, peppermints amongst a really big grove of grass trees. They're looking a bit rough and ready there. They're resprouting post-fire, but the peppermints were looking very, very sick. That site was sprayed with phosphite, and the red area there was part of the Wild Otways Initiative that our friends at Forest Fire Management and DECA helped us deliver. The red area was sprayed with phosphite at 12 kilograms per hectare. There's one line where we sprayed the spoon drains, and the red area is that big block to the right hand of Egan's Track. And that this will actually be one of our treatment sites. This is actually that exact same area that you saw beforehand after it's been sprayed with phosphite at 12 kilograms per hectare. Absolutely looking fantastic, and we were delighted at the response to the peppermints because they were looking very, very sick there, but the grass trees look great. I'm moving my cursor around a yellow coloured grass tree in the background. That's currently dying from Phytophthora. We couldn't save that one. However, that site will get another treatment as part of our program. And this is what it looks like. To the right-hand side, we've got the catchment for Lees Creek off Egan's Track. To the left-hand side, we've got Sandy Creek. Phosphite will be applied to the red square, which means it'll have a couple of treatments of phosphite and over the last couple of years. The blue square to the north will be a new treatment, and the rectangular shape to the right will be another treatment that will be conducted as part of this program. Boulder Hill in Western Australia, the left-hand pink-coloured triangular shape is where phosphite has been applied every second year since 2012. That will be an important focus site for us for this particular project, particularly with Jeff's work on mycorrhizae. To the right-hand side is a pink square that's near the King Creek on the south coast of WA there, and that will be our treatment for this year. So both of those sites will form part of our treatment, and they'll be both treated with 12 kilograms per hectare of phosphite. We have vegetation surveys. A lot of the work on plot-based surveys has been very powerful in Western Australia. We don't need to focus too much on the floristics there, and they will look mainly at phytotoxicity, which they haven't observed anyway. 1 to 4 hectare plots for treatment. WA, we've got to get a wriggle on late March to early April, but thankfully our scientists have started their permit applications there, which is good. Victoria, we're looking to apply in May. New South Wales and Queensland, ideally July and August respectively. And the management objectives we're talking about there, particularly for Eastern Australia, for sites from Queensland through to Bruny Island in Tasmania, where we're seeing dieback affecting habitat, not just plant species, but habitat. And it's the habitat of species like smoky mice, New Holland mouse poecula, Kangaroo Island dunnart, and a whole host of others. So that's where we're at. Coupled with this is the statutory review of the Threat Abatement Plan. And Barb and myself have helped coordinate some of the feedback there. The traditional owner feedback that we've received on the current plan has been compelling, and we certainly have made a number of recommendations to our colleagues in DQ about that particular feedback, so it'll be interesting to see how that goes. And I'm going to call it quits, having chewed up most of your time.
Thanks, Mark. That was fantastic. We've got lots and lots of questions in the chat, but and I'll throw to Andrew to address those. But one comment I'd make is that, you know, it's a great example of how research, working with land managers, working with regulatory authorities, and ultimately, you know, helping hopefully form a bit of a policy and a pathway forward for addressing cinnamon fungus. A great bit of work, well done. Andy, the one or two big ones that are popping out in the chat, and then we might have to call it quits. Thanks.
Mark, you've done a great job answering a lot of questions already in your slides, so I'm going to leave those aside. First question though, are you happy to share your slides with us? A lot of people are interested in having as a reference. We'll send them around as part of the seminar, email group. Question about the susceptibility of plants. Do we know why some species are more susceptible than others to this pathogen? And is there any evidence of in
a crude sense, some species have the ability to wall off the pathogen at the fine roots. So they just produce a barrier, and the hyphae of the pathogen can't get in. So that would be a crude sense. Those plants that can't do that get infected by the pathogen. They've got an increased ability to do that when they're sprayed with phosphite.
So are we seeing any plants develop that as a natural resistance? Like, are we getting selection for species that within a population?
It's been looked at to get dieback-resistant jarrahs for forestry in Western Australia. A lot of investment there, very marginal, very, very marginal. This pathogen is an absolute doozy, and it can hide in an already impacted area for many decades as chlamydospores, and when other susceptible plants start to try and recruit, it takes them out again. It's unbelievable.
So given that it can hide away, how often do you need to apply phosphite?
Excellent question. WA 5 years. WA 2 years. We think it's got a lot of legs, and some research scientists would think it's got a lot of legs. We reckon we can probably get a good management program with 5-year treatments on tracks in Victoria every 5 years.
That's encouraging. Cool. In terms of the interaction with fire, what happens in the landscape with fires coming through and cinnamon fungus?
We haven't measured it closely. Our observations from the Boggy Creek bushfire in 2020 in the Otways was post the fire, the disease bubbled. The consensus with specialists around Australia is that when we get post-fire expression of the disease, it is very, very aggressive. So our recommendation is if we're going to spray phosphite, even if there has been fire, it is worth it. And I think that that is true of what they have done in the Stirling Ranges. I'd be keen to see whether or not we might be able to do that at Chapel Vale in the Otways, but I need to talk with my dear colleagues down at Colac about that.
Okay, thank you. And final question, I'm going to squeeze it in. There's a comment around phosphite possibly increasing phosphorus levels in the soil. How do native species cope with that change?
It's been measured with, again, Sarah Barrett, Dion and co leading those studies on areas where they've sprayed phosphite for 30 years. That was just published in late 2024. No measurable increase in soil, plant available soil phosphorus.
Thank you so much, Mark.
Good
effort getting speed questions.
Great. That was like a speed dating exercise there. So thanks again, Mark, for a really great presentation, and also to Barb for your presentation. 2 excellent examples of how science is informing policy and management. I don't know, I don't want to commit on behalf of you guys, but there are a number of other questions left in the chat. So that might be something that we could bundle up and send to you to see if we could get some sort of response from you after the fact. Appreciate that we've gone a bit over time, so apologies everyone for that. But I just want to say thank you for attending today, and just to say that this is the first, of course, of our monthly ARI seminars for the year. Our next one will be on the 24th of March with a focus on saving our aquatic species. So we've got some presenters from ARI, but also the Victorian Fisheries Authority on that one. You can also find out about our seminar recordings on the ARI website. So Tess is putting all that in the chat, and we're always keen to hear feedback about how we can do things better and help get this important science out into the community, and to help inform decision making and create impact. So if you've got some thoughts around that, then we're very happy to hear them as well. But again, thank you to Barb and Mark for 2 excellent presentations today. Thanks everyone for attending, and thanks for Andy and Tess working behind the scenes. Have a good week all. Thank you. Bye.